All. This is Dr. Mobeen Sayed from drbean.com. Welcome to one more show. So, if you hear my nose a little congested, my apologies. I have been under the weather for a few days. Okay, so Chit Chat started. Quick reminder that Chit Chats will move to the other channel, the KoolBeens Cafe Live, uh, in another two Chit Chats. So, let me share the link here so that you can um, become a member there or subscribe there. And so we'll move to that. What I'm doing at this time is that when we do the chit chat, I leave it up till Monday and then I hide it and I put the link on Patreon. So Patreon can continue to watch it, but we generally hide it from the YouTube because there is not much viewership after the weekend time. So this is one. Now, today I have a couple of very interesting things for you. One, there is a breakthrough therapy for type 2 diabetes, insulin dependent type 2 diabetes from China. And that is that they have actually created cells, beta cells, from a person's own blood cells. And those cells have worked. And the person who was insulin dependent is not insulin dependent anymore and has left all the medicines for diabetes as well. This therapy for with stem cells has been researched for years and decades, but I'll show you a couple of uh, type of therapies and inform you how the Chinese therapy is a little more advanced. The second thing that I want to do today is that I have written an article about glycation. So I thought I'll quickly go over that with you. That way I can present glycation once again to you. Plus, there is some structure in that article. It's not published yet. It's just in my documents. And so we can look at that together. And finally, of course, we'll do chit chat. And a quick housekeeping thing. Mark has been really kind that he has been curating the questions that you ask here, which I have not answered. And these are tons of questions. The question now is, how do I answer all of them? Do we take a chit chat to answer them? Do we do additional talks to answer those? These are a lot of questions. And so that I do not know what is the right answer there. Okay, so with this, welcome everyone and happy almost about the weekend. Let's start with our discussion. And of course, if you have a question, please ask that with some cues in the beginning so I can scan that and quickly see where the questions are. So this is drbean.com. And if you would like to get access to more lectures, just visit drbean.com and become a member there. Teresa Ramagas is saying, make a video. It is a lot of hard work to put videos together. I want to make sure that the questions are not just using my time in abundance, because the questions are in abundance. So we have to find a solution that is better than this. Maybe what we can do is people become members of Patreon, and then their questions that are here, I can answer them and upload them in Patreon, or as we discussed in two Zoom sessions on Wednesdays, live. Maybe they can ask those questions there, or we can answer them there. We'll have to figure something out. I can't give so much time that the whole day long I'm just busy creating videos for answers, because the questions are a lot. Okay, so here is drbean.com. I'm going to start from this one. The links are in the description. So this one is breakthrough by Shanghai doctors uses stem cells to cure diabetes. So before we understand what this is, I made some diagrams for you to kind of create a context for understanding this kind of therapy. This is not just the only therapy. There are other studies and researches that use this technique. So When we are developing inside our mothers, the very first cells, the sperm and the ova that fuse and then they start making the first fertilized eggs, these eggs would give rise to something that is called totipotent or omnipotent cells. Why these are called totipotent? Some people call it totiopotent. So whatever is the right pronunciation, I call it totio because the word total is used. Totiopotent or omnipotent cells. These cells are interesting. Imagine those the sperm and ova fuse and then they divide a little more and they make a cluster of cells. This cluster of cell is important and interesting because these cells are capable of making the embryo, the human being, and all the structures around the embryo, the placenta and the embryonic sacs and various other structures that are necessary for that growing embryo to implant in the uterus, to connect with mother and to receive and exchange nutrition and waste products, use her oxygen, etc. So totipotent cells are able to make not only the human being, but all the structures around it at the embryonic time. So we say that these cells are capable of making embryonic tissue and extra embryonic tissue. Then these totipotent cells will give rise to multiple kinds of other cells. Of course, for example, these cells are going to give rise to a cell type that would make the embryo. They might give rise to another cell type that would make placenta and so on. So the next cell type that they make are usually called pluripotent. Pluri from plural. So the pluripotent cells are also stem cells. Stem cells have a quality that they can give rise to cells that will become something else. Plus, they can replenish their own pool as well. So this is a definition of a stem cell, that it has to be able to renew its own pool. It has to be able to make more stem cells. And then it makes further other types of stem cells. So here, this is pluripotent. It is not omnipotent. Why is the pluripotent interesting, or what is the role this is playing? Pluripotent cannot make embryonic and extra embryonic tissue. These cells will give rise to cells that will only make the human being. These cells do not have the capability of giving rise to cells that will make placenta or other things. So you can say that they are a little more restricted in their capabilities. They are smaller in their capability as compared to totipotent. But still, for a human being, all of our cells, their daddy cell or mother cell, are the pluripotent cells. These are the cells that give rise to every other kind of cell. So these can give rise to any germ layer cell. What does that mean? When we are developing, we actually develop as 3 sheets of cells. If you take 3 papers and put them on each other, lay them flat on each other, these are the 3 sheets of cells from where we develop as a human being. And of course, when we are developing in the form of a sheet, we are a 2-dimensional structure. And then slowly the structure would start wrapping around. It would wrap like this from side to side and give us a structure. It will wrap from, above downwards to give us a structure. So those sheets will fold in various ways to finally make a 3-dimensional structure that we are. So these 3 sheets are called germ layers. So the pluripotent cells are able to give rise to cells that make up various germ layers. And why do we care for different kind of cells? Because each germ layer can make a different set of body tissues. For example, this germ layer, or ectoderm as we call it, makes the skin, the outer layers of the body. And then there is mesoderm, and then there is endoderm. Endoderm will help make cardiovascular system. Mesoderm would help make, for example, GIT and so on. So various cells in the germ layers will make various kind of tissues. And the pluripotent can give rise to all those cells. So as I said before, they are kind of the granddaddy of all of them. Here is where the technique they used. What they did was, and you would see this for many other researches as well. What they did was, so let's see if I can, they took blood from someone. In the case of Chinese study, they took the patient's blood and they took some cells from there. For example, they took T cells and the B cells and the natural killer cells. Excuse me. They took those cells and then they manipulated their DNA. Interestingly, so think about this with me for a second. It's really interesting. Interestingly, our cells that are present throughout our body, they have all the DNA available in them that was present in these stem cells. So what is interesting is that a muscle cell is keen on just making a muscle cell, or a muscle cell's DNA only functions to operate as a muscle cell. But it has all the genes to be a pluripotent cell. So what happens is that as cells start committing, we call this committing, that the pluripotent would say, I'm going to make a cell and this cell will become pluripotent. The totiopotent would say that. And the pluripotent cell would say, okay, I'll be a pluripotent. I would never try to be the totiopotent again. I'll just be a pluripotent. So how does that happen? Some of the genes in this cell become locked. These genes will not open again in this cell's life. These genes that are locked or even removed or kind of locked, they were the ones that were responsible for creating capabilities of this cell. So now as this cell, the pluripotent, start giving rise to more restricted cells, their genes are further locked all the way down to a fully differentiated cell. Differentiated cell means more graduated cell to a specific function. For example, a muscle cell is a fully differentiated cell. It has become a muscle. It is like a baby who has graduated to be a doctor, let's say. So let's say a muscle cell or a blood cell actually still has all the genes of the pluripotent cells as well. It's just that those genes are locked away. So the scientists, what they did was they kind of opened up those genes, or they added those genes back to these blood cells. So of course, now the blood cell, let's say this is a natural killer cell, and you open the genes for the pluripotential, then that blood cell would convert into a pluripotent cell. It will become a stem cell. I remember when this breakthrough came, that it's a normal cell could be taken up and manipulated to become a stem cell again. I was just so excited. I think it was 2006, near that time. I was excited because if you can make the stem cells again, you can then make anything from there. So the idea of somebody losing a liver or a pancreas or a kidney can actually be, in theory, fixed, that pathology, by just making a new liver or a new kidney and so on. Although there is a lot of complexity in that, and this team from Shanghai kind of overtook that complexity. So what they did was they took the blood cells and they coaxed them. They actually treated them to become pluripotent cells. Such cells are called induced pluripotent stem cells, or iPSCs. And this is again a technology that has been now used for some time. So once they were able to make pluripotent cells, then they could coax those cells to make pancreatic cells. So let me go down this line for a second. So we have pluripotent stem cells. Then the pluripotent stem cells start giving rise to other cell lines. And each line of cell promises to make a specific kind of tissue or system. For example, let's say mesenchymal stem cells will make a lot of structures that come from mesenchyme, for example, bones and cartilages and muscles, etc. So these are mesenchymal cells, which would give rise to these mesenchymal structures. Similarly, there could be a cell line formed who will promise to make blood cells. And that would mean that this line, this stem cell will promise that, hey, I promise to go and make a T cell and a B cell and a red blood cell and a megakaryocyte or a platelet, etc. But this cell would promise that I will not make anything else. I would only make blood cells. I won't go and say, I want to make a muscle cell. This cell over here, who has promised that I will only make mesenchymal cells or mesenchymal cells, this cell would say, I'm not going to go and start making blood cells. I commit to only making muscles and bones and cartilages, etc. So you can see that as more and more daughter cells are coming in, they are becoming more and more restricted in their capabilities. This process is called differentiation. They are becoming more and more differentiated. You can think of this as babies as they start growing. They become teenagers and they have an idea of what they'll become. Then they go to 20 years, 21 years, and they are studying in a school and going to become something. And then they become 25, 6, 7, and they have become an MBA or a doctor or an engineer or someone else, right? So these cells are committing or differentiating into specific lines of work. So these are called multipotent cells. Multipotent cells can give rise to a set of tissues, but not all the tissues. For example, hematopoietic stem cells will make blood. Hematopoietic, they are heme or blood making cells. Mesenchymal cell will make these structures, as I said before. Then imagine that we have hematopoietic stem cell. That parent cell takes on the responsibility of making all the blood cells. So what it will do is it will make children who are also stem cells, but now they are called oligopotent stem cells. Oligo is one, just one potency. For example, maybe this stem cell can only make T or B cells. Maybe another stem cell can only make red blood cells or myeloid cells. Meaning, as they keep coming down the line, they become more and more restricted in their function. Finally, when we reach very close to the tissue, final tissue, we can encounter unipotent stem cells. For example, within our muscles, there are stem cells that can make more muscle cells. So they are unipotent for that. They have one potential only. And they will not make anything else. Although they are stem cells, they can replenish themselves too, but they could only make a muscle cell. And of course, then let's say at the end, there is a muscle cell. So we will say a muscle cell is fully differentiated. It has become stable. It's not a stem cell anymore. It is a functional final cell. A red blood cell is a fully differentiated cell. A bone cell is a fully differentiated cell. A GIT gland cell is a fully differentiated cell. They have committed to being just that. However, you can pick them up, open up the genes, and make them walk back. this line and come all the way and become pluripotent again. So this is one technique that they used. Second thing that they did was even more difficult. And that was to now tell this cell to become a pancreatic cell. So they were now going to coax this cell to walk it down this line once again, all the way down to the pancreatic cell. So first they had to encourage this pluripotent cell, and I'm using the word encourage. They would put various kind of growth factors and various kind of chemicals that are released in us. Think about this for a second. When we are developing, we are nothing but a cluster of cells. How does some cell know that I'm going to go somewhere in this abdominal area and make a pancreas there? Or I'm going to go further down and I'll go and make a kidney there. Or I'm going to go further down, I'm going to go make, urinary system or genital system. Or I'm going to go even further down and make a foot. And they know where to make a foot and where to make a kidney and where to put the pancreas. How do these cells have their own internal GPS to know I am in the right place to make a pancreas? So because I am here, I'm going to make a pancreas. And then that whole process of a cell to reach the place where to make the pancreas and then actually differentiating into a pancreas, replicating that process in the lab to take a pluripotent cell and say, man, you're not going to make all three germ layers. I just want you to make, let's say, mesodermal layer, because the pancreas comes from the mesodermal layer. So I want you to make the mesodermal layer. So first, this has to become qualified, differentiated to mesoderm. Then within the mesoderm, we have stomach and intestine and pancreas and tons of other things. Now you have to coax him further, or it further, to say, you know what? Go to the area where the pancreas is made. Get into that mindset. So that cell says, okay, fine, I'll become a multipotent system which will make all the tissue of the pancreas. Then they say, okay, very good. But no, we don't want you to make the whole pancreas. We just want you to make an islet. Islet is the structure within the pancreas in which beta cell and alpha cell and gamma cells and delta cells are sitting, not gamma cells, but delta cells. Then they tell this one that, hey, become more specialized to only make the islet cells. And then from the islet cells, make the pancreatic cells, the beta cells, for example. And when those cells are formed, then they take those cells and put them back in the patient. Now, what is the interesting part in this technology is the following. If you see this study here, or this news here, this is Harvard, October 20, 2021. I remember that the very first time when the stem cell was created from another somatic cell, meaning another, I think they did it from a skin cell. It was in Japan. So here is Harvard. Harvard is saying a new therapy for treating type 1 diabetes. And if you look at this article, this is exactly the same process that they took some, they took some cells and they converted them into pluripotent. And from there, they coaxed them to become pancreatic cells. The only thing is that this one was not patient's own cell. There were some cells. Because of that, when they will put these cells in the patient, some patient, they will have to give immunosuppression. Otherwise, those cells will get rejected. Right? So, of course, this patient actually felt better and they had to immunosuppress him so that those cells do not get rejected. On the other hand, I'm going to give you an aside. And I have been talking about that when I was doing this COVID discussions. I believe I had always wanted to create a project called Project Phoenix, where we re-emerge from our cells. And how do we do that? When we are babies or when we are younger, just like nowadays there are placental banks. Imagine that a child gives a biopsy of their skin, their pancreas, their liver, their kidney, their heart, their blood vessel, and that is stored away. And as the child reaches the age of 50 and 60 and various tissues start failing, you go to your bank and you get your pancreatic cells, which are your cell. That is a donation of your cell back to you. And you just grow them and put them back. And now you got your own new pancreas, which will give you another 60, 70 years of function. Similarly, you do that for liver, you do that for kidney, and you donate your own tissue for your own self. So I wanted to make this company for a long time. I used to call it Project Phoenix, that will have a tissue bank. Anyways, from that aside, if I come back here, this is what they did, but they did better. Instead of somebody donating their own tissue back in the childhood, what they did was they took this patient's own tissue, own blood. They manipulated that blood back towards pluripotent cells. From there, they then developed in incubators with proper growth factors and proper transcription factors on the cells. They developed back the pancreatic cells, the beta cells. Now, these beta cells are patients' own cells. So when they put them back in the pancreas, nobody is going to come and reject them. Unless it is a type 1 diabetic who may have a problem that their immune system is attacking their beta cells. In that case, if it is a cell, type 1 can be multiple pathologies, not just the cell. But in that case, they could actually change the cell so that it does not become the target of the immune system. But here it was a healthy person. So they just took his cells, made them into beta cells, put them back in his body, no immunosuppression needed, and the patient recovered. So if you see here, Jin Hao, a leading researcher on the team and director of the hospital's organ transplant center, said they use the patient's own peripheral blood mononuclear cells. These are T cells, B cells, monocyte, natural killer cells, etc., and reprogram them into autologous induced pluripotent cell. So here the word autologous or autologous, whatever way you want to call it, is very important. Autologous is my own cell. So these are patients' own cell. You didn't have to fight with the immune system to say, leave those cells alone. So into autologous induced pluripotent stem cells, they used technology they devised to transform them into seed cells and reconstituted pancreatic islet tissue in an artificial environment. So here the seed cell, those stem cells that were kind of unipotent type stem cells or oligopotent type stem cells, they're simply calling them seed cells. And then further coax them or provided them 3D environment that these cell thought we are in the body of a developing embryo and we are at the place of making a pancreas and we should go and make a pancreas. And not only the whole pancreas, we are in the place of making islets and we should make islets. They regenerated that whole environment for them. And the cells became pancreatic islet cells. And that is how this technology worked. So the result is that this patient, within a few months and years, first did not need insulin anymore, then did not even need the drugs anymore like metformin. So he has become fully healthy. So Jean says that, where is my mouse cursor here? I remember you talking about your brain. Thank you very much. I've been talking about it for a long time. So this is a very interesting technology. This is actually very interesting therapy. 2024 5/13. So a few months ago, they declared it. In my opinion, we should actually put more resources on these kind of technologies and rid people of diabetic outcomes. I can assure you that just controlling diabetes of a person will give them health, neurological health, muscular health. The whole body will become refreshed. Glycation would go away, tissues would start becoming better, they would operate better, and the person would be new. So, this is one technology I wanted to share. Now, this is the KoolBeens Cafe. So, if you are going to search for this on YouTube, KoolBeens is one word. KoolBeens Cafe Live. And from a couple of more talks later, our live chit chats will only be here and not on this channel. Although I found out that I could actually just hide the video within a couple of days and then leave the link on Patreon, so people still have access to it, but they are not on the YouTube channel, and YouTube is not trying to promote them and people not viewing them, and YouTube saying, well, I do not think that this is a great channel, and not penalize it. Paul says, I hope the Chinese are not cheating again in their papers. Wouldn't be surprised if they found it easier to go back to the omnipotent stem cell, grew a whole human, harvested the cell. It is, I can't say, but really, as soon as you can go back to any of those cells, if you can go back to a pluripotent cell, you can make a new human being. Yes. If you can go back to a totiopotent cell, then you can actually implant that cell back into a mother and she would develop a baby. Okay, so next. This is the chapter I was writing. There are some diagrams in here that are not mine. I have just put them as fillers to say I need to make diagrams in these areas. And this is about glycation. So I have a question for you. Do you want to discuss glycation today and look at this chapter, or you just want to do chit chat? Either way is fine. If you say chit chat, I'll just stop sharing and we'll talk. If you want to see this chapter, then I'll go over this with you. Your call. Margaret says, perhaps you can help with a similar program here. I could. Margaret, I am not as credentialed for any of the government bodies to work with me or other research teams. I am a simple doctor and a software engineer. That's all my credentials are. Okay, so Paul says, let's talk. Let's talk. Aim says, chit chat, how to calm down. Okay. How to calm down is such a subjective question. The reason is we all have a different way of calming down. I'll give you an example. In the past, I had an accountant. Great guy, Michael. I will not. Great guy. Meaning he just did so much. He helped me so much throughout my many years. And sometimes when I would, for example, let's say something is submitted incorrectly in taxation, I'll call him and I'll say, I did this. What should I do? And or let's say HR block did something. He would say, go and yell at them. And I'll say, why should I yell at them? He'll say, that would relax you. He used to, cut jokes. So he'll say, that would relax you. You'll yell at someone, you'll feel better. And I'll say, no, yelling at someone would actually make me even more nervous and tense. So how to relax is everybody's own learned behavior. And that is taught to you from your upbringing. However, the outcome of relaxation is common in everyone. And that is the stress hormones need to go down. The corticosteroids need to go down. norepinephrine needs to go down, epinephrine needs to go down. Now, you could do certain things that would generally relax you. For example, I said this in the past. If you take a pencil and you put it in the mouth like this, it kind of forces you to assume a smiley face. Even if you're depressed, even if you're sad, if you just assume a smiley face, your brain thinks that you're smiling. Brain systems are not telling each other what they're doing. So the smile system, when that becomes activated, the personality system doesn't know that I did not ask to smile and this person is just faking the smile. The personality system would actually think that, oh, we are smiling, and it would actually make you happy. On the other hand, if you take a pen and hold it like this, the pursed lips, that is actually a for brain, that is a signal that you are sighing and you are not happy, and it actually makes you tense. So many times we have this habit of at least I had it in the, remember the joke, I went to this post office in COVID time and I licked the pencil accidentally and I got COVID. So sometimes if we do like this, we actually make ourselves more nervous because of the way we have our expression. Similarly, if you, let's say, develop tachycardia or palpitations, one can massage one side of the angle of the jaw, not both. Both can actually faint us because the carotid bodies are here. So if you massage a little over here, that not too hard, not too much, just a little, and there are proper techniques for the carotid body massage, and you should learn that from your doctor or from nurse or whoever. Doing a little massage here actually very quickly reduces the heart rate and the palpitations can be controlled. The reason for that is that carotid body is sitting here doing blood pressure monitoring towards the brain. So if you massage here a little, the carotid body doesn't know that the pressure is not from the blood, it is from your hand. It just thinks that, well, there is going to be higher blood pressure in the brain, and it would ask the brain to ask the heart to reduce the force of contraction and number of beats, and that reduces palpitations. Similarly, I believe that there are so many psychological methods to become better or relax. Another important one is being near the plants or trees. The oils that trees emit, if you can smell them, these oils are getting into you. And they have done, this was a Japanese study, I have mentioned it many, many times, that in Japan, they take two groups of people, they gave them off, this gave one group money and sent them to the city and asked them to go have fun, have party, relax, do whatever. And the other group, they sent them to forest and said, just be there for 2 weeks. And before these groups went, they had checked their natural killer levels, they had checked their cortisol levels, their stress hormone levels. So they did a complete immune system and stress system profile. And so you would think that the people who went to, who had a lot of cash, who were loaded with money and went to the city and were partying there, they will probably come back more relaxed. It turned out that people who were next to the plant oils, when they came back, their natural killer cells were performing better, their immune system was performing better, and their stress hormones were significantly lower compared to those who went to the city. So being near the plants actually relaxes you. And if you cannot be near the plants, then there are those plant oils that you can put lamps at home and they can give you fragrance. I have plants here, so you can have plants at home as well. So that is another way to relax and calm down. And then there are happy places. People can imagine their happy places and go there and calm down. So there are so many ways to calm down, but it also depends upon what is stressing you out. Sometimes what is stressing you out overrides all other aspects, all these techniques. And in that case, you need someone with you. And I think one one dark side of depression is that a depressed person does not want to get help. They sometimes they just don't want to call someone, and they're not even that um, happy, or they're not that engaged. But calling on a friend, family is a very important thing as well. And of course, then professional help as well. Exercise helps a lot, absolutely helps. It changes the whole environment of the body. It takes away it moves the lymphatic system, it moves the muscles, it moves a lot of lymphatic system and the CSF as well. So brain fluids become refreshed, muscles become refreshed, dopamine is released, endorphins are released. Endorphins actually make you happy. So exercise is a very good way to relax and calm down. So Andy says that how did they isolate the plant oil as the helpful agent? So I think that one, they had a theory already that the plant oils would do this. All these studies come from some sort of a hypothesis. So they may have had a hypothesis before, and then based on that they did this. Ames, thank you very much, Ames. Faraz says separation of wood oils. Ames says thank you, you're very welcome. What else? A. Perry says walking outdoors on woods or prairies is a great relaxation for relaxer for me. Absolutely. Muhammad says terpenes, trees are magic. Yeah. Brett says smoking is a mental break. And again, that is a learned behavior. Forest bathing is amazing. The morning light is amazing. Evening light is amazing. So for us, says, can pressing carotid cause syncope and death? It can cause syncope if both sides of the carotids are pressed. But the thing is, you can't hold them hard enough that you just have a hard blockade. But of course, this is also a problem when people have a noose around their neck. But generally, just one side of massage is decent enough. Guy says, watch The View and you'll know why we need Viagra. Okay. Danielle says, if cancer can be starved as a metabolic treatment, is there any other ways to neutralize the cancer directly? So, Danielle, I want to correct this because I hear this from a lot of people, and they can end up causing people to become to die. Not all cancers can be just that easily starved by not eating food. So, cancer starvation is a very specific kind of a therapeutic approach where, excuse me, where you have to starve the cancer cells and not the remaining body. And so it is not as simple as let's just start fasting. So, but there are so many ways nowadays that we have started approaching cancers. Brett says that I would disagree that smoking is just a learned behavior. Nicotine has an effect on us. Nicotine is a different thing. So if you said nicotine affects in a specific way, then I would agree that I would not say that's a learned behavior. But if you said smoking helps, then smoking is a learned behavior. To give you nicotine, not only that, there are some psychological things attached with smoking as well. Old school says, I didn't miss all of it. No, only the important part. John says that, John, if it is a question, please cue there. You had a recent discussion on ABI. Remind me, ABI, which term, and I would discuss further. Aim says, thank you for answering. You're very welcome. Okay, so what else? Old school says, oh, well, I'll rewatch. Thank you very much. My apologies. My voice is a little different, but my whole face and throat is congested. And this is once again Cofix. Whenever I travel and I use Cofix, I do not catch anything. And whenever I don't use Cofix thinking I'm fine and there's going to be nothing, I catch something, and then I just kick myself to say, why was I not using Cofix? It actually happened. I traveled 3 times in last few months. And the first time I didn't use Cofix, I became sick. The second time I learned my lesson, I used Cofix and I was fine. And the third time now, when I went to Los Angeles and came back, I didn't use Cofix, and here I am. Farah says, have you covered not being calm and hair loss? No, I have not, but there is hair loss can occur because of stress as well. Yes. Cliff Finkel says, crow can't beat cat. Yeah, so Luffy's meowing outside. Let me see if I can open the door and he can come in. So 1 second. Luffy, come on in. Okay, here is Luffy. Luffy? What happened? What happened? Somebody bother you? Something happened? Don't destroy my mic. Okay, all right. So that was Luffy. Let's see. Daniel says, is there any study that shows impairment of mitochondria by COVID infection? Yes, there have been many studies. And it is actually mitochondrial dysfunction can occur for so many. Mitochondria are kind of very delicate systems, and they would, under inflammatory state, they can become damaged very fast. And once they become damaged, it is a problem to recover them, especially if the tissue itself is compromised. So if I can give you just a few ideas. Mitochondria has electron transport chain in it. I remember I did a study about COVID and mitochondria where they said that as COVID enters the cells, the ACE2 binding and the spike proteins production within the cell causes certain enzymes to become activated that cause mitochondria to stop. Some of the mitochondrial enzymes reduce their activity, which in turn cause mitochondria to produce less ATP. That is its job, to make energy. When the mitochondria makes less ATP, but there is a demand on ATP, mitochondria in turn starts producing inflammatory markers to say, I am under stress. I'm not able to run as I should have, while there is a demand for me to run. And so it would change its state and it would start producing inflammatory markers, cytochrome C and the other, even mitochondrial DNA can spill out. Reactive oxygen species would start getting emitted. So what happens is the electron transport chain, we have complex 1, 2, 3, 4, and then ATP synthase, right? So these are 5 proteins that are structured, and there are millions of such factories running within the mitochondria. If the mitochondrial enzymes aren't functioning correctly to be able to run this machine in the presence of oxygen and nutrition, in the presence of the demand that is present, then what happens is nutrition is present, energy is being produced by the, let's say, complex 1 and 2 and 3, but the complex 4 is not working correctly. The ATP synthase is not working correctly. So this energy that is backing up in the factory would start escaping as reactive oxygen species. The result is, first of all, mitochondria itself will get damaged. And I used to give this example when I used to teach mitochondria and SARS-CoV-2. Imagine that you have a iron factory where you have hot iron that is passing through an assembly line. And then it goes from various stages, and then at the last stage, it becomes something, or you store it, or you do something with that. And imagine that the last stage is not working. It's not receiving the iron, hot iron, but the first stages are still pumping. They are melting the iron and they are pumping it towards the last stage. What will happen is, last stage is off. The first stages are now pumping the iron. There is no place to pump it. They will become overwhelmed, and the iron would start becoming spilled out in the environment. That's exactly what happens, that you have the nutrition, the complexes are not working correctly, electrons are present, energy is present, it's backing up in the system, and it is in the form of reactive species. It's very high energy state in the mitochondria. Now those species would start getting spilled out when they'll be the reactive oxygen species. Things like CoQ10, vitamin E, vitamin C will try to neutralize them. And it's a very interesting thing. CoQ10 is the first one to neutralize these reactive oxygen species. CoQ10 is a very potent antioxidant. However, imagine I am a CoQ10 and I pick up some reactive oxygen species to neutralize it. In that process, I would change to, I believe it is called ubiquinone. I'll become ubiquinone. I am now in a reduced state. I have gotten this energy and I'm holding on to this, and I'm like saying, oh, what do I do with this? Vitamin E and vitamin C has to come and take that energy from me and convert me back into ubiquinol, I believe, or my normal CoQ10 state. If vitamin E and C themselves are overwhelmed or used up by these reactive oxygen species, the hot iron that is running around, then there is nobody to pick up CoQ10 and pick up this heat, that the hot iron that it is holding. So in that process, the CoQ10 itself will burn down. So what happens is, and this is true for many inflammatory states, many mitochondrial dysfunctional state, but COVID does this very potently. So what happens is that the mitochondria just becomes so damaged that the reactive oxygen species, the fireworks starts within the cell, then the cell starts becoming damaged, and the whole, all bets are off. Cliff Hinkley says, nice cat, thank you very much. So Brett says, if nicotine didn't have a beneficial effect and only the act of smoking did, we would be smoking cherry wood. Sure. I think what you're trying to argue with me is that the smoking is not a learned behavior, but somehow it is an inherent quality in us. I would let you be with that concept. I am saying something else. AJ says, question, is Glenn Jeffries in London College infrared red recharging mitochondria theory? So I do not know about Glenn's theory, but I can tell you this, that infrared actually helps. Methylene blue and infrared help mitochondria recover unless it is too much damaged. And what do I mean by too much damage? Excuse me. Imagine that this is a mitochondria, my room. In this mitochondria, there is DNA of the mitochondria, there is RNA of the mitochondria, there are enzymes of the mitochondria, there are electron transport chains that are running on the membrane of the mitochondria, millions of those chains. There are shuttling mechanisms that are bringing in nutritional molecules. Oxygen is coming in, then it is being used, ATP is being formed, ATP is being released back into the cell, carbon dioxide is formed, water is formed, and all that machinery is running. If for some reason the electron transport chain, the fireworks making machine, becomes dysregulated, these fireworks that would go off, the reactive oxygen species, they will be immediately caught by CoQ10, by vitamin C, by vitamin E. But if the production of fireworks is faster, then when these three things can actually neutralize them and recover from them, then those fireworks, the reactive oxygen species, would start damaging other things. The first set of damage would occur to the DNA of the mitochondria, RNA of the mitochondria, meaning within the machine of the mitochondria, the damage would start occurring. Then the reactive oxygen species would leak out into the cell as well, and they would start damaging the cell too, the cytochrome and these organelle there. That means a mitochondria may be dysfunctional and may be able to recover back towards health, or it may be damaged sufficiently that it cannot recover back. The whole machine is broken. So let's assume for our discussion that mitochondria is in a state that it can recover if you provide it help. So what kind of help can we give it? There are many ways mitochondria become healthy again. One way is, imagine that this mitochondria, and this is a mitochondria, this mitochondria is kind of not very healthy. Some parts are damaged. Both mitochondria can fuse together. And now they become one big mitochondria. This is mitochondrial fusion. It would allow the healthy machinery of the healthy mitochondria to be used by the remaining machinery and electron transport chain of the unhealthy one, and both together would start becoming better. Similarly, it is possible that there is a mitochondria that is unhealthy, and maybe there is a little damage over here. That mitochondria can decide to split into two. And one is the unhealthy one, and one is containing most healthy material. This is called fission, mitochondrial fission. And now this one is healthy and this one is not healthy. And this one can wither away. Or under stressful state, healthy mitochondria can do fission and make more baby mitochondria that would grow and become bigger. And now we have more energy and ATP production happening. These are all health maneuvers. One more health maneuver is near infrared light. Just like plants have this green chlorophyll in them, and the behavior of the chlorophyll is that it can interact with light, correct? Mitochondria has the complex 4. The complex 4 has cytochromes in it. These are chlorophylls in it. Not chlorophylls, but chromes in there. These are metals that can interact with light, and they can absorb energy from them. So when you put infrared light, when you beam that on a mitochondria, near infrared light can penetrate up to 4 inches through the clothes and the bones, and it can enter the mitochondria. When it falls on complex 4, which is a cytochrome containing complex, which is in layman term, chlorophyll containing complex that can use energy to do its function and create energy. The cytochrome, the complex 4 would kickstart itself. It would start working with the energy coming from the light. And when it would start working, the complex 4 can now utilize nutrition, it can utilize oxygen, it can actually pump protons, which would then activate the ATP synthase. So just these two, complex 4 and ATP synthase, in the I hope I am unfrozen by now. So if you can hear me, these mitochondria, who, let's say, the electron transport chain complex 1, 2, 3 are not working correctly, 4 was not working correctly. You gave it near infrared light, 4 became active. Now it is consuming oxygen, it is producing carbon dioxide and water, it is consuming electrons and stuff. Plus, it is directly taking energy, photons from the light. It doesn't care that the transport chain 1, 2, and 3 are not working. When it is using that, it is pumping protons from inside to outside. Those protons will go to ATP synthase and go back inside, and the ATP synthase would run and ATP will be produced. So from a 5-set chain, just 2-set chain is running. And that is what near infrared light can do to mitochondria. So a sick mitochondria can start working. Now, when the sick mitochondria starts working, guess what happens to the cell? ATP production starts. Cell has gotten energy again. Cell starts building its systems again. Cell starts functioning again. Mitochondria knows that I have a pressure to make energy, and I'm making energy. So the mitochondria's inflammatory markers that it is releasing that I'm under stress are gone. Mitochondria switches back from inflammatory state to healthy state, which causes the remaining chain to start working again as well. The cell becomes healthy again as well. When this happens, the story doesn't finish here. When this happens, the complex 4 has nitric oxide attached to it. Now, there are many, many theories in literature about this. Some theories say that the nitric oxide is actually produced outside of the mitochondria. It arrives to complex 4 because it modulates the production of ATP. So it influences complex 4. There are studies that also show that complex 4 holds on to nitric oxide as a modulator of the vascular diameter control for that tissue. What does this mean? I hope I'm not frozen. Am I talking to myself? Am I unfrozen? Margaret says better. So I hope you can hear me. So, okay, Aim says you never froze. Okay, unfrozen. Okay, so where was I? I was talking about the story doesn't finish there. When the mitochondria starts functioning again, yes, when mitochondria starts functioning again, it would start releasing nitric oxide from complex 4. Nitric oxide, we all know, its job is to do vasodilatation. Levoarginine is used as a supplement to do vasodilatation in people who have vascular inflammation. Viagra type things, they help with vasodilatation. It is actually said that taking nitric oxide improves the erectile function because vasodilatation occurs. When a sick tissue on which you beam near infrared light and its mitochondria start becoming better, they start releasing nitric oxide, which opens up the blood vessels. And now this sick tissue would have more blood flow through it. Guess what would that do? Bring in more nutrition, bring in more oxygen, wash away more waste products and carbon dioxide and acids and creatinine and phosphates. The whole tissue would start becoming healthy. Just how? By beaming near infrared light on the cells. So that is how beautiful the effect of near infrared light on sick tissues is. Again, it would depend how damaged are the mitochondria, how damaged are the tissues. But within reason, this recovery can occur. Paul says, I've had good results from near infrared light. Excellent. Excellent. Yes, so near infrared can be very, very useful. The only thing, if somebody is going to use it, near infrared, 10 to 12 minutes, not lesser than 10, not more than 15. Number one. Number two, there are many lamps that are available on Amazon or other, which are mixed lamps. That is, they have near infrared and infrared, or thermal light and less thermal light. A few chit chats ago, I made a comment that near infrared only raises the temperature by 3-4 degrees, while the thermal light can raise temperature a lot, like saunas. Then later in that chit chat, I said near infrared is cooler or cold. And I thought that it would be caught that it is relatively cold compared to thermal light. So near infrared is warming light as well. It is energy as well. However, it raises the temperature slightly. So if you are going to buy a lamp, make sure that it is not mixture of thermal and near infrared, because the thermal light or heat producing light or heat conveying light has a different way of working and different time need. It should be used for more than 15 minutes. Near infrared, lesser than 15 minutes. If you turn on both of those lamps together, any one of them will become useful and the other one will become a waste. So if you buy separate lamps or switchable lamps that you can switch back to near infrared. And if you don't want to buy a lamp, morning walk, evening walk, when the sun is at an angle, you got more infrared light at that time, which can penetrate through your clothes and get into your body and help with the healing. Faraz says singular of mitochondria is mitochondrion. Thank you, Faraz. Faraz says occasional very brief freezes. So it happens here in my area near 6:30 is that there are some freezes. Maybe people just turn on TVs. So this is a Danielle says, how do cancers? This is such a huge topic, but I'll just talk about one mechanism. This is a huge topic because the cancer cells are just so hardy cell that they evade and escape immune systems punishment. They can turn off the immune system. They can pull all the energy. They can waste a lot of energy. So one thing that the cancer cells do is they use a mechanism which I actually taught on Dr. Bean last week. And I would request you to become a member there because I just taught hypoxia inducible factor last week. And this is part of the inflammatory pathways lecture. And learning those pathways is important for all of us. So in our cells, we have a transcription factor. Transcription factor means a protein that will go into the nucleus and manipulate genes. There are many transcription factors. Excuse me. One of them is called hypoxia inducible factor. What that factor does is that when a cell is under hypoxic state, less oxygen state, it teaches the cell to survive that state. How does it teach it? So that's the discussion I did for 1 hour last week. But generally, it goes to the nucleus and opens up certain genes that make certain changes to the cell's functions. One of the change is metabolic change, where it switches the cell from oxidative phosphorylation to glycolysis. For the oxidative phosphorylation, we need oxygen. And if, let's say, a cancer cell was going to use oxidative phosphorylation and the cancer cell's environment is such that there is hypoxia in there, the microenvironment for the cancer, then the cancer cell would die. But when the hypoxia inducible factor becomes active and it teaches the cancer cell to use glycolysis, which is anaerobic breakdown of the glucose and making of the ATP, and it is made even more efficiently as well, but lesser in quantity with more waste products, the cell starts surviving. Now, I think in glycolysis, you take one glucose molecule and make 2, 3, 4 ATPs compared to oxidative phosphorylation, where you make 32, 36 ATPs. So the cancer cell starts surviving. They start using an anaerobic respiration or the glycolysis. Plus, they waste a lot of energy because now they're taking up glucose and only making 2 ATPs versus 36 ATPs. So their demand for energy increases. So they have figured out a way to survive and use the energy, the nutritional substances to make ATP. But in that process, they need a bunch of energy to be able to survive and continue to work. This is the background to which then certain doctors say we should starve the cancer. The problem is you can't just start starving yourself to say, I'm going to starve the cancer. Cancer would always pull the energy from the body before the other cells can. So that means if you eat less, the remaining healthy body would suffer more than the cancer cells, because cancer cells have activated the mechanisms to pull the energy and use it fast. So one has to be really careful about that. But this is one way that the cancer cells know how to survive and live in harsh environment. A. Perry says, good to know about the sun angles. Correct. So the morning when sun is rising and the evening when sun is setting, it's not very hot. That is when the percentage of infrared light that we get from sun is more than the thermal light. So these are excellent times. If somebody cannot buy a lamp, hopefully they can go out and walk in the morning and evening and get a bunch of near infrared. Okay, what else? What is the next topic? AJ says, I've been using 670 nanometer for retina morning treatment. It has helped for my vision. Yeah. So from 600 to 700, 800, even 1000, they say is near infrared. I would say 600, 750 is a good range. Your friend says, Some studies show high doses of antioxidants may reduce ROS too much, increasing cancer risk and reducing exercise benefits. How can we find the right balance? So I'll have to look at that study to see what is the too much of the antioxidant they're talking about, because generally our body is able to decide, turn on and off various supplements, their usage, their cofactoring based on the environment within the cells. So I have to see what was it in vitro versus in vivo, because in vitro the cells are sitting all alone. They do not have any surrounding environment to help them at all. And secondly, how much was the dose? That will have to I'll have to check that up, because it seems like an incorrect general statement. But if it is in a study, there must be a backing up data, and I need to look at that. Faraz is selling another YouTube channel here. So I have heard this too many times, that doctors claim on YouTube, actually, not doctor, nutritionists and some, the fitness channels, don't take too much omega-3, don't take this. It really depends what is that study and the environment of the study. This is why whenever I speak about something, I back up with some study that I have gone through and looked at it. I completely dislike and disrespect those individuals who just make up their opinions and just keep providing that without saying this is my opinion, and they have some sort of data which is not really going to back up their opinion, but now they are just saying their opinion and saying I have a study to show this. Snowbird says, isn't fasting effective against cancer? So once again, Snowbird, it depends. What cancer, what is the stage of cancer, what is the microenvironment of the cancer? Understand this, that the cancer has, if it is growing, it has created a microenvironment which is acidic, which is hypoxic, and which is able to, by angiogenesis, able to pull a bunch of nutrition and blood flow from us. Saying that we can starve that cancer by just starving the person is such a horrible uh, position to take. The reason for that is very simple. It's mechanical reason, not theoretical. Cancer cells have changed their model to use glycolysis. They have changed their environment to work with acidic environment. Excuse me. They have created bunch of blood vessels into them to pull preferentially the blood flow. So they are sitting at a privileged state that first food goes to them. How much can we starve a person? Because when you start starving someone, that would cause less. So let's say the food, if somebody was taking 3,000 calories, now they're taking 1,500. Cancer is still going to pull a proportionally greater amount from that, and the rest of the body will receive less, and that would cause a problem. The high metabolic state cancers, there are certain cancers which, when you see under imaging, they take a lot of energy very fast, and specific types of energies or nutritions. You can manage them by starving them of the kind of nutrition they are looking for. But that has to be cancer to cancer, person to person, and which cancer is pulling what nutrition. That cannot be just a generalized statement that he has starved the cancer. So this, Daniel, isn't the correct state. This is actually an incorrect. The cancer cells are able to work with glycolysis much easier than oxidative phosphorylation. And it's a very simple thing. I just talked about this switch. The cancer cells have hypoxia inducible factor activated, HIF activated. HIF's function is to switch the metabolic processing from aerobic to anaerobic. So it's just a very different way. So I don't think it can be generalized. Brett says that after all the money that has been put toward, and I'm sure there was a time when cancer diagnosis was a certain death certificate. Nowadays, there are many cancers that are being handled very well. And I have done 2 or 3 videos on cancers where even those cancers that had reached the stage 4 and they had told the patients that you only have a few weeks to live, and then they did some exotic therapy and they are now the cancer free. So we are making good progress. I think it can be accelerated further. We'll see. Brett says 3,000 to 1,500 isn't starving, to zero is. Sure. So, Brett, I hope you have some credentials to talk about how energy systems work in our body. When we are starving, when we are fasting, let's say zero, you ask me to stop eating today. Body will first of all start pulling energy from fat tissues. Once it has eaten away the fat tissue, then it is going to start pulling energy. So even before the fat tissue, first of all, the autophagy would start. Microautophagy would start, then macroautophagy would start, chaperone-mediated autophagy would start. After that, fats will be used. After that, muscles will be used, and then any other tissue that can be there. So thinking that somehow if you make a person absolute zero calorie, would somehow the cancer would say, you know what, I am stopping, I can't work with this, while the remaining body would just continue to function, is not a correct understanding. Faraz, I do not know. You have to provide that. So your friend, I'm going to look up this study, antioxidant supplements promote tumor formation and growth. Okay, so let's see if this is a study. We can look at it together. Antioxidant supplements. So this is 2021 December. Promote tumor formation and growth and confer drug resistance in hepatocellular carcinoma by reducing intracellular loss and induction. So the patient is already cancerous. Now let's see, is it in vitro or in vivo? Controversy over the benefits of antioxidant supplements in cancer persists for long. So this is the specific case here, is that it is usage of supplements within cancer patients. Using hepatocellular carcinoma as a model, so this is an in vitro study. We investigated the effect of exogenous NAC and GSH. Multiple mouse models, including, so this is kind of in vivo animal model. Multiple mouse models, including this model induced this model, hepatoma cell and human HCC cell, xenograft model, okay. In vitro assays, including Ross assay, colony formation, sphere formation, okay, so they did that. In these multiple different mouse and cell line models, we observed that NSE and glutathione promote HCC tumor formation and growth accompanied with significant reduction of intracellular reactive oxygen species levels. So the question is, what is this level? Moreover, NSE and GSH promote cancer. And when I'm speaking about question is, I'm not challenging it. I'm kind of asking, this is how I read studies. That, okay, so what do they mean over here? So you are seeing me reading a study for the first time with my thinking aloud process. Exogenous supplementation of NSE or GSH reduces the expression of NRF2, suggesting related antioxidant production pathway might be desensitized. Using transcriptomic analysis to identify potential gene targets, we found that TMB1 BIM1 was significantly upregulated upon energy treatment. Both in-house RNA sequence database showed that it was a tumor. Okay, so they're saying that when we gave ROS, when we neutralized ROS, then this gene became more active, and this is overexpressed in tumors as well. Stable knockdown of this increased intracellular ROS. It also abolished the promoting effect of antioxidants. On the other hand, BSO and SSA inhibitor. Okay, so that is the conclusion or data implicates exogenous reducing intracellular. Okay, so in cancers, the question is the amounts. Full text link. I mean, if you take out reactive oxygen species, so there is a genetic disease where the neutrophils cannot make reactive oxygen species. And those children, they develop repeated infections, even to the point of they dying, because they just cannot handle the infections or pathogens, certain kind of pathogens. Of course, they will not be able to handle cancers because reactive oxygen species are used for us to handle these kind of things. So imagine that this study brought the levels down as if there is no reactive oxygen species. Then yes, infections would happen, cancers would happen, the person cannot survive. Antioxidant supplements promote tumor formation. Let's see. Design. Material. NSE, GSH, and this were obtained from sigma cell lines. These were the lines. Dose equivalent. High doses of NSE. So 120 milligram per kilogram per day. So 120 milligram per kilogram per day. So imagine if we have an average person of 70 kilogram, 120 multiplied with 70, 8400 milligram. NAC RDA. I'm just going to say dose. 600 to 1200 milligram per day. So if you take 600 milligram per day, they're giving 84, let's say 1000. So they're giving 8 times more NAC. Then what did they do? Glutathione 100 milligram per kilogram per day. So if you take a person of 70 kilograms, so 100, so 7,000. Now what is the glutathione? Glutathione dose. 250 to 1000 milligram. So both of those are given at 8 times. Given to them were equivalent to 600 for a 60-day kilogram human adult. 120 milligram per kilogram and 100 given to the animals were equivalent to 600 milligram tablet daily, 10 milligram and 500 milligram tablet daily for a 60 kilogram human, as calculated with the FDA recommended conversion. 12.3 for mouse from human equivalent. Okay, so they are saying that they are giving these as standard human doses. Then the evaluate tumor formation. So according to this study, then what I'm seeing is that if a patient of cancer is taking ROS killing things or antioxidants, it can compromise their cancer fighting capabilities. So at least this is the study in a first blush. The question is, did they give any clinical guidance or did they say more studies? One second, just going to the end of it. And it actually makes sense that why would we want to kill the reactive oxygen species in a cancer patient? Because he's using them to fight with cancer. So in the current study, we found that tumor effect, of course. Now, this tumor promoting is when there is a tumor present. Conclusion. Our study showed that exogenous antioxidants, NSC and glutathione, by reducing the intracellular ROS levels and inducing this expression, promoted HCC formation and tumor growth, and counteracted the therapeutic effect of the drugs. Inhibitors that target NSC and GA metabolite reverse these. Okay, so this study does show this, that if there is a patient of cancer and you take their antioxidants out, even with the standard doses, then their cancer, their cells will not have sufficient firepower to fight with the cancers. So thank you for the study. Good study. Katin says, how are you feeling? Much better, still a little congested. Good to see you again. You're a great teacher. Love, light, and truth. Thank you. Same to you as well. Andy says, did Paul Merrick talk about? I think Paul has this belief as well that cancer can be starved. I do not believe that. Brett says it is frustrating that only those who have approved credentials can be listened to. That's not the point. The point is, I do not have enough time to go through the whole mechanism. And when some other folks inject statements in people's minds to disentangle them, I'll give you this example, Brett. A few days ago, there was a internet presenter who presented a talk of about 10 minutes, 9 minutes, in which he said that if you have pitting edema, take these supplements and you'll be fine. Just that 9-minute talk and taking only 30, 40 seconds of message took me whole day to research and hours to explain why his message could cause people to die. So there is no offense. It's not about that. When somebody is credentialed, my hope is that when we walk about certain things, we can understand. Otherwise, I just have to spend a lot of time to go step by step to explain. For example, take this message of starving the cancer. So let me explain what I'm visualizing in my head. Cancer does angiogenesis. Cancer activates hypoxia-inducible factors. Cancer moves away from the normal metabolic behaviors of the other cells. Cancer preferentially pulls food and nutrition and oxygen from the environment. It already starves the other parts of the body. There is a thing called cachexia that happens with cancers. Not only it releases chemical substances that tells the other cells to not eat, but it also eats their food. And so the body starts becoming, shriveled. for such a situation, starving the person. I mean, look at my situation, that if I sit here and says, go starve a person of cancer and he'll be fine, that's horrible. Starving a person cannot be a standard um, statement, generalized statement. There are certain cancer that are very high in their metabolic needs. You could starve them in theory by doing their excision of blood vessels or taking out a bunch of their material, etc. But if you starve a human being, then cancer still gets the first call on the nutritions, and the remaining cells would become even more hungry and damaged. This discussion, so for example, here, Warburg effect, this discussion actually needs, so the discussion of the Warburg effect and the discussion of those mechanisms need a lot of attention to go through those things. I can't just simply either agree with you. If I do that, I'm making a mistake. And to explain why I don't agree, I would need 3 hours. Correct. So Faraz says that phagocytes use peroxide to kill. Absolutely. Neutrophils do it. So if I go back to my sharing my screen, it is actually such an interesting thing to, there is actually a test for, so if you do oxygen burst test. So measurement of oxidative burst in neutrophils. So neutrophils use reactive oxygen species or reactive species to actually burn, excuse me, to actually burn the pathogens or offending agents. And there are tests which would take a person's blood and they would take the neutrophils, and then they would see if those neutrophils are able to produce reactive oxygen species or not. And if they're not able to produce it, they cannot handle infection. Such people actually live in a very difficult situation because they keep getting repeated infections because they do not have enough ROS, or their neutrophils do not have enough ROS. Phoebe says that thanks, that makes a lot of sense. You're welcome. Brett says that thank you for taking your time. I've never seen this site. Thank you. I did not mean any offense, but some of the KoolBeens here who were with me when I, in my Zoom talk, in my frustration that this person said in 9 minutes things that I would need to take 3-4 hours to explain, and I had to verify my own knowledge. I became so gaslit because that person has millions and millions of subscribers. My video takes in a month, this chit chat would take what, 3,000 views? He gets millions in a week views. So imagine his range, his influence, and the damage he did this by just that one video or 30-40 seconds long message within that video. And so this is why it becomes a little hard. And those KoolBeens who were parts of Patreon or Substack, they were with me, and we were going over point by point to say, look how wrong this person is and how dangerous he said. And it took a lot of time to explain it. I felt gaslit. Then I spent the whole day confirming that I was right, and what I thought he was wrong was wrong. And he says, and there I was thinking Ross was, no, Ross. So anytime our body need to dismantle something, burn something, fight with something, it uses Ross as the weapons. So that is our main weapon. Final common pathway of destruction for viruses, bacteria, fungi, worms. Although worms, it's so funny that, worms, we have these eosinophil cells. I always think of them as they do 1,000 cuts to kill them. So worms are big things compared to the little cells. So the eosinophil cells, they come and they surround the worm, and they start releasing little acidic things which cause cuts on the worm skin, and the worm start leaking fluids. This is just like if our skin gets burned, we start leaking fluids and we just cannot survive if the percentage is a little bigger. So these tiny cells cause 1,000 cuts on the worm's surface to kill it. So there are many other ways too, but reactive oxygen species is a very important part with which our cells help kill cancerous cells, kill pathogens, kill damaged cells, etc., or destroy them or dismantle them. So Sigrid says, is there a difference between feeding the body ketone versus glucose in relation to the cancer? So I know that there are theories that if we switch to ketones, then the cancer cells cannot use that kind of nutrition. Yes, this is true that cancer cells convert to glycolysis, but they are still our cells who know how to work with various kind of nutritional substances. So again, this is why I'm kind of saying it again and again. There are specific type of cancers which have nutritional demands of special types, and you could try to starve them. But even for them to be starved, you will have to really plan how to do it to protect the person and starve the cancer. Either take out the part of the cancer or cut its vessels, then you try to do other things to it. So Sly Fox says, when would you have your friend Susan Oliver on your channel? It doesn't have to be a live stream, and you could tell her that. So actually, I talked with Susan afterwards as well, and I was joking with her that my some actually just you, some folks have asked for Susan to be there. She said, "Yeah, I can sit down and do a recording," but I don't. I mean, she does her recordings. I do my work. There is no common thread at this time that I need her to come in, but she's fine. Not live, recorded. If there is some interesting topic, I'll invite her. But she's okay with that. Okay, so almost 2 hours. I hope I did not waste your time. I have to say thank you to your friend for this study. It was an interesting study. Good study. Okay, so looks like we are good. I don't see any more questions. Aim says you helped us a lot. It was my pleasure. We were all going through a really rough patch, so we all had to do whatever we can to support each other. Paul, you're very welcome. We learn from you as well for stats. Oprah, fun Oprah. You're very welcome. Brett says thank you, and you did not waste it. Thank you, Brett. Your friend, thank you. And thank you, your friend. Sigrid, thank you. Aim, thank you. Stephanie, thank you. Perry, thank you. Garner, thank you. Phoebe, thank you. Sigrid, thank you. And with this, I'm going to go. The door is open. Cats are out. I'm going to go find the cats and bring them back. You have a nice weekend. I would leave this up for a couple of days. On Monday, I will hide it. So if you have the link, you can always watch it. And I'll post the link back on Patreon so they can watch it if they like. Bye-bye for now. Happy weekend. And tomorrow, 7 a.m. Pacific time, we'll have our Zoom morning walk. The link must have been published on Substack, Patreon, and on YouTube. Bye for now.